I think once you have worked with long-read DNA sequencing, you cannot live without it. So, of course, if I started today, I would initially start with long-read sequencing immediately.
The data that we generate from African genomes needs to be shared with the global community. Rare diseases is a team sport, and we want to be part of that team. And we want to be sure that patients in Africa ultimately benefit from the discoveries that emanate from our research.
In population scale research, you have to be ambitious. You have to set big goals and try to deliver something that's of benefit to as large a proportion of the population as possible.
So I think a sustainable, scalable model of genomic medicine in Africa starts with data ownership. I think that's a really critical part of the life cycle. And so what I mean by that is that, for example, in our work, we really prioritize the generation of HiFi data in country. This enables us to start with smaller sample sizes. It also empowers us to build capacity around the analysis and interpretation of HiFi data. And later, as we think about clinical applications and diagnostics, we would be investing in the expertise to actually take that forward.
I would definitely recommend to younger scientists to look into easily implementable genetic findings, such as pharmacogenetics or monogenic findings with high impact that are not too rare. This would definitely give them an easy place to start.
My advice would be to try and link up with others in the same field who perhaps have a similar perspective and similar experience, and to connect with those individuals to try and build a network on the continent to support each other.
I want to collaborate with many different countries in projects such as HiFi Solves and learn from each other internationally of how we could drive genomics into the clinic faster and better together.
So I think in terms of collaboration, we want to be equal partners in these kind of efforts. We don't want to, necessarily just be sample collectors and send DNA to large consortia. We actually want to be part of the process from the bottom up because we want to learn and we want to grow capacity. And so I think, partnerships that acknowledge that and that share our vision would be really helpful.
I think the first thing other countries can say is definitely look at Estonia. If they can do it, why can't we? So once we actually did the comparison and saw what kind of data we got out of long-read sequencing and then just the quality of the data that we got from the PacBio sequencing, we were convinced.
We do have the capacity available. We've got the human capital to develop, and we do have, for example, the computing resources to do it. I think it's just about connecting all the pieces of the puzzle to make it happen. We are admittedly behind in the genomic revolution, but I also think that gives us a very unique perspective and chance to really evaluate what's going on in the field and adopt the current best practices, which might be very different from what was done, say, 20 years ago.